Hi students, in this module we are going to discuss about testing of bio-nano packaging materials. In the previous module we discussed about green plastics and bio-nano composites and have learned that green plastics along with the bio-nano composites provide a better alternative for synthetic food packaging material. We also learned that bio-nano composites helps in improving the properties of green plastics and make them near perfect for food packaging applications. In this module, we are going to discuss about what are the properties of bio-nano packaging which makes them perfect for food packaging applications and also discuss about methods of testing these properties. In this session, again we are going to have a detailed discussion on different properties of bio-nano packaging material and their method of testing. We discussed in several places the two major problems which are associated with the use of synthetic polymers in packaging applications are utilization of non-renewable resources as a raw material for their production and the non-biodegradation nature of the packaging material itself. These two major disadvantages of synthetic polymers paved a way for several renewed interest in the use of biopolymers as packaging material. Students, do you know how these biopolymers are classified? Well, they are classified into two categories. The first category includes biopolymers which are derived from naturally occurring materials such as proteins, cellulose, starches and their polysaccharides. The second category includes the one which are chemically synthesized from the naturally derived monomers such as lactic acid. Now, some of you may have a doubt whether these polymers are perfect for food applications. The answer for your doubt is a big no. I mean to say that the biopolymers too have many limitations in terms of their poor mechanical, barrier and thermal properties when compared to synthetic polymers. Then you may ask how to rectify this. Is there any solution for it? Now, the answer to your question is a big yes. Dear students, in this juncture, I would like to introduce a new term to you, that is the bio-nano composites. Yes, you please understand that the use of bio-nano composites along with the biopolymers drastically improves the properties of biopolymers and make biopolymers as a perfect alternative for synthetic polymers. Then what are the bio-nano composites? They are the nanoparticles of 1 to 100 nanometer at least in one dimension. That means in the lengthwise or breadthwise or the depthwise. They are incorporated into the biopolymer matrix to get bio-nanopolymers of improved and desired properties. Now, the question before you may be what are the desired properties of food packaging material, so that we can also expect the same type of properties in the bio-nanocomposite food packaging material. If you look at the properties, it includes biodegradation properties, mechanical properties, barrier properties, thermal properties and finally the rheological properties. How to test these properties? Testing these properties is very important in assessing the performance of bio-nanocomposite packaging films as food packaging material. Let us discuss the importance of these properties with respect to food packaging applications and the method of assessing or testing these properties. Methods and standards for assessing biodegradation process. The extent of biodegradation of degradable plastics under different environments such as marine, soil, sewage and compost depends on the species and genera of microorganisms present in these environments. Depending on the ability of the microorganisms, biopolymer degradation may ranges from the complete degradation to negligible degradation. Consequently, for a biodegradable polymer to be used for a certain application, the biopolymer should necessarily degrade in the environment of end use. Evaluation of biodegradability should consider the end use of the biopolymer and the environment of the end use. The following tests and methods as well as the standards protocols proposed by the American Standard Testing Method, often called as ASTM, and the Organization for Economic Cooperation and Development, often called as OECD, are used to assess the biodegradability of the polymers. There are several tests. These tests includes modified stun test, closed bottle test, petri dish screen test, environmental chamber test, and finally the soil burial test. Let us discuss about these tests one by one. What is there in the modified stem test? This test operates under aerobic conditions. The test substances provides the sole source of carbon. It is exposed to a low level of inoculum, and a non-specific method of analysis is used to follow the course of biodegradation. This test usually runs for a period of 28 days without any acclimatization period. The process of stun test includes addition of test substances in two concentration, that is 10 and 20 milligram per liter, to a chemically defined mineral nutrient solution which is free of organic carbon, followed by the addition of inoculum of sewage microorganisms, that is in the range of 1 to 20 into 10 power 6 ml per liter. Then the test system is with suitable control incubated at ambient temperature with stirring for 28 days. Carbon dioxide is evolved during the process and it is trapped in an alkali and converted into carbonate. This carbonate is measured either by titration method or by a carbon analyzer and calculated as a percentage of total carbon dioxide which the polymer could have theoretically produced. Now the second method for the degradation assessment is closed bottle test. In this method, predetermined amount of test substances is added to a chemically defined mineral salt solution. The solution is then inoculated with sewage microorganisms and then dispersed into closed bottles. The bottles are then incubated in a dark condition at a temperature of 20 plus or minus 1 degree centigrade and periodically assessed for their dissolved oxygen content. The oxygen demand is calculated and compared with the theoretical or chemical oxygen demand of the test substances. Well, the next method of assessment is using the petri dish screen. In this test, the principle involved is the testing material is placed on the surface of a mineral salt agar in a petri dish containing no additional carbon sources. The size of the material must be 2.5 by 2.5 centimeters square, and this test are approved by several developed countries such as USA, German, French, Swiss and international organizations. In this test, the mixed inoculum of known fungi or bacteria are then sprayed on the test material and on the agar surface. The amount of growth is observed on the surface of the petri dish after the incubation period of 21 and 28 days at a constant temperature. A rating was given based on the growth for assessing the fungal resistance. More growth on the surface revealed that the material is instinctively able to support the growth of the microorganism and thus leads to the greater degradability of the polymer. The fourth method of assessing the biodegradability is the environmental chamber method. In this method, also a visual assessment of amount of growth on the test material is made and a rating given for assessing the biodegradability. Test procedure includes hanging the strips or prefabricated components of the test material inside an environmental chamber of high humidity, that is usually around 90%. High humidity environment encourages the microbial, in particular the fungal growth. Standard mixed inoculum of known fungi is then sprayed on the test material and incubated for a period of 58-56 days at a constant temperature. Then finally, the soil burial test. Soil burial test is yet another test for assessing the biodegradability of the packaging material. This test, based upon this technology, evaluate in-service soil contact exposure. The material is buried in the soil bed prepared in the laboratory, commercial soil-based compost. The soil beds are normally conditioned for up to 4 weeks period to use and may be supplemented with organic fertilizer to encourage an active microflora. A cotton textile strip is used to test the microbial activity, which should lose 90% of its tensile strength within 10 days of the exposure to the soil. The moisture content is normally set around 20 to 30%. Samples are removed for assessment. The change in the sample surface and nature of microbial growth are identified using light microscopy and SEM images. Next important property of polymer is mechanical property. So far we discussed about the tests for assessing the degradability of the biopolymers. Now we are going to discuss about what are the mechanical properties required for the bio-nano packaging material. The mechanical properties such as tensile modulus, tensile strength and percentage elongation at break are the important properties of concern for bio-nano composite based packaging material. Tensile modulus is the resistance offered by the material for deformation, whereas the tensile strength measures the ability of the material to withstand maximum tensile stress. On the other hand, the third property, that is the percentage elongation, represents the flexibility of the packaging material of concern. Now let us discuss these mechanical properties. First we will discuss how tensile testing is done. Tensile testing is done according to the approved method of ASTM D88202 or ASTM D63803. While tensile testing of thin sheets of less than 1 mm thickness is done using ASTM D88202, ASTM D63803 is used for the sheets of 14 mm thickness. Universal testing machine is commonly used for tensile testing. In this method, rectangular specimen is fixed in between the movable and stationary fixtures. Movable fixtures moves at a constant velocity till the sample breaks. So if you look at the details in figure 1, basically the figure 1 is obtained by plotting the applied load on x-axis and the resulting elongation of the packaging material on y-axis. The curve thus obtained is called as force elongation curve. The important mechanical properties includes tensile modulus, tensile strength and percentage elongation. Tensile modulus is represented as Tm and is calculated using the following formula. Tm is equal to del F divided by A suffix I whole divided by del L divided by I suffix I, where del F and del L represent corresponding changes in force and length during the initial linear deformation. Ai initial minimum cross sectional area of the specimen, Li initial gauge length of the specimen in between the grips of the instrument. Now the tensile strength. The tensile strength is termed as Ts and is calculated using the following formula. Ts is equal to F suffix B divided by A suffix I. In this, Fi represent force at break point, whereas Ai represent initial minimum cross sectional area of the specimen. Now the third property, that is the percentage elongation, is mentioned as percentage E and is calculated using the following formula, that is percentage E is equal to L suffix B divided by L suffix I into 100, in which LB represents elongation at the break point. Well students, now we will discuss about test for the barrier properties of bio-nanocomposite packaging material. You very well know that the shelf life of a food product is highly influenced by the barrier properties of the packaging material. Knowledge about the barrier properties helps to understand the bio-nanocomposite materials resistant to sorption and diffusion of gases and moisture across the packaging material. Improvement in barrier properties of the bio-nanocomposite material is achieved due to the tortuous path created by uniform dispersion of the nanoparticles in the bio-nanocomposite film. Water and gas molecules passing through the tortuous path would obviously take longer diffusion time, thereby always showed an improved barrier properties. Barrier property of the bio-nanocomposite film is measured by water vapor permeability, often represented by WVP, and oxygen permeability represented by OP. Now let us see water vapor permeability. Water vapor permeability is defined as the rate of water vapor transmission per unit area of a flat material per unit thickness induced per unit vapor pressure difference across the material. Water and desiccant method are the commonly used method for measuring the water vapor permeability. This is the method prescribed by ASTM E9605. In the water method, a relative humidity of 100% is created and maintained inside the test dish by distilled water. Water vapor loss from the test dish is measured periodically to determine the water vapor loss from the test dish to the controlled atmosphere. While in case of desiccant method, calcium chloride is used to seal the test specimen kept on a test dish to maintain 0% RH inside the test dish. This entire step is housed in an atmosphere of known temperature and relative humidity. Water vapor transmitted through the test specimen is measured by weighing the test dish at certain time interval. Now the water vapor transmission rate. Water vapor transmission rate and water vapor permeability are calculated using the following formula prescribed by the ASTM standard 2005A. Water vapor transmission rate is calculated using the formula water vapor transmission rate is equal to capital Q divided by small t into capital A, where Q is equal to change in weight in terms of kg, T equals to time in terms of minute, and A represent the area of mouth of the cup in meter square. From the calculated water vapor transmission rate, water vapor permeability is calculated using the following formula. Water vapor permeability is equal to water vapor transmission rate into del x divided by del p, where del x represents the thickness of the test specimen and del b represent vapor pressure difference across the test specimen. Now let us discuss about the another barrier property, that is oxygen permeability. Oxygen per unit area of a flat material per unit thickness induced per unit vapor pressure difference across the material is measured as oxygen permeability OP. It is measured by hanging the test specimen in between two chambers under ambient conditions. Of the two chambers, one is filled with nitrogen, that is acting as a carrier gas, and the other is filled with oxygen. Oxygen permeates through the test specimen into the chamber with nitrogen. Carrier gas nitrogen carries the oxygen to the coulometric detector, and an electric current is produced and is directly proportional to the amount of oxygen flowing into the detector per unit time. This is measured as oxygen transmission rate. Oxygen transmission rate, that is OTR, is calculated by ASTM standard 2005B formula as OTR is equal to V suffix E minus V suffix 0 into Q divided by A into R suffix L, where V suffix E represents steady state voltage level, V suffix O represents 0 voltage level, Q suffix C represents calibration constant, and A is the exposed area of the test specimen, and R suffix L represents the value of load resistance. The calculated oxygen transmission rate is used to measure the oxygen permeability again by using the formula given in the ASTM standard 2005B as OP is equal to OTR into del X divided by P, where del X represents the thickness of the test specimen and P represents the partial pressure of the oxygen in the oxygen chamber. Well students, so far you have learned different types of tests used for measuring the various properties of biodegradable polymers. Now I will move on to the next important property of the bio-nano composite packaging material, that is thermal properties. Among the thermal properties, class transition temperature, thermal stability and heat deflection temperature are the important thermal properties of bio-nano composite packaging material when they are intended for food packaging applications. Differential scanning calorimetry, thermogravimetric analysis and dynamic mechanical analysis are commonly used proven methods for measuring the thermal properties. Now let us see what is class transition temperature. Free volume theory, that is volume available for a molecule for its free internal movement or relaxation time, is used to describe the class transition phenomena of a polymer material. Free volume is directly proportional to the temperature of the product. During class transition states, movement of the bonds between such as bending and stretching as well as movement of side chain occurs. This transition corresponding to bending and stretching of bonds is known as gamma transition. Beta transition is associated with the movement of side chains and is related to the toughness of the material. During further increase in temperature of the polymer, free volume also increases drastically, facilitating the large scale movement of polymer chains. It is known as alpha transition or class transition, and the associated temperature is known as class transition temperature. At this temperature, amorphous solid changes from glassy, that is relatively brittle state, to the rubbery state, that is a softer state. In addition to this state change, class transition temperature is also responsible for the abrupt change in the specific heat, coefficient of expansion and storage modulus of the polymer film. Dynamic mechanical analysis and differential scanning calorimetry are the two main techniques used to determine the class transition temperature of the polymer. Let us see what is heat deflection temperature, HDT, how it is affecting the polymer property. The temperature at which a polymer deforms under a specified load is measured as heat deflection temperature. It is an indices which measures the ability of the packaging material to withstand heat to an applied load. Commonly used method for measuring the heat deflection temperature of a bio-nano composite is ASTM D64807. In this method, maximum stress of 0.455 megapascals or 1.82 megapascals is created at the center of a rectangular cross section test specimen using 3-point bending column. The temperature rise of that specimen is recorded during the experiment. The temperature at which the test specimen deflects by 0.25 mm is identified as heat deflection temperature. Finally, it is a rheological property of the bio-nano composite packaging material which determine the processability, that is the melt processing behavior of a bio-nano composites during unit operations such as injection molding and blow blow film, etc. Important rheological properties include steady shear measurements, dynamic shear measurements, and extensional shear measurements. Now, I will be discussing what is steady shear and how it is measured. Rotational or capillary rheometers are the commonly used instruments for measuring the steady shear measurements for bio-nanocomposites, while parallel plate or Conan plate geometry rotational rheometers are suitable for low to medium range shear rate measurements. High shear rate measurements are done using capillary rheometers. The flow behavior in injection molding process is well described by high shear rate measurement. Next important rheological property of concern is dynamic shear. The microstructure and morphology of bio-nanocomposites may get distorted by steady shear measurements technique, thereby reduces the accuracy of the result. Henceforth, the dynamic shear measurement is recommended for the examination of the microstructure of bio-nanocomposites. In this technique, as a prerequisite, the bio-nanocomposites are subjected to small deformation. As a mandatory procedure, dynamic shear measurements of the polymer material must be carried out at its linear viscoelastic region, as a reason that the viscoelastic behavior of the polymer material is independent of deformation at this region. As noted by the renowned scientist Bhattacharya and others during 2007, stress relaxation, grip recovery, and dynamic oscillatory deformation are the other parameters that could be studied to understand the dynamic shear characteristics of a bio-nanocomposites. Finally, it is the extensional measurements. During the process of film blowing, blow molding, and injection molding, bio-nanocomposite polymers are subjected to extensional flow apart from shear flow. Often, this behavior of the polymers are expressed by their extensional viscosity. Extensional viscosity is measured by measuring the resistance of the polymer while the polymer is subjected to stretching. Mincer type rheometer and continuous drawing of micro monofilament are the commonly recommended method for measuring the extensional properties of the bio-nanocomposite polymers. While the earlier measures the transient extensional viscosity, the later measures the extensional rheology. After a very long discussion on testing of bio-nano packaging material, I would like to conclude that the environmental pollution caused by the use of synthetic packaging material reached a dangerous position, particularly in developing countries. This could be mitigated by the use of biodegradable polymers. Study of biodegradable polymers is a newly emerging field. The study focuses on the improvement of some of the important properties of the biodegradable polymers, such as mechanical and barrier properties with respect to packaging applications. Bio-nanocomposites technology is one of its kind which could improve the properties of biopolymers drastically, in addition to reducing the cost economics of the biopolymers. Bio-nanocomposite technology could able to do so by increasing the aspect ratio and surface area of the nanoparticles. Several standard tests are available to measure the properties of biopolymers, which we already discussed in detail. Thorough understanding about various testing methods is a prerequisite to manufacture biopolymers with desirable properties, which makes bio-nano packaging material a perfect for food packaging application.